Okay, so this first video is going to be the bacterial smear. We'll do it separately from the gram stain. So first of all, I have my protective gloves on, which are going to help me because obviously some of the specimens that we are using might be harmful. So here is my specimen in the petri dish, also known as ketchup in this experiment. And first what we're going to do is basically we want to get a sample of the specimen onto the slide so that we can stain it and then view it under the microscope. So this is going to be my grease pencil, and we're going to use this so the ethanol when we're staining doesn't wipe it off. So what we're going to do first is on the slide we can write proper name of the organism. So for example, if it was E. coli, we could write E. coli to make sure that we use the universal name and everyone knows what it is. And then secondly, we can draw a small circle, and that's where our sample will go. And then we will flip it over so that we are not putting our sample on the side that we wrote on. So next what we're going to do is we are going to use our inoculating loop, and we're going to use the aseptic technique to make sure that nothing is contaminated. So here's our Bunsen burner. So we're going to hold it over the Bunsen burner for 10 seconds, and that will help to make sure that the inoculating loop is entirely sterilized. 8, 9, 10, okay? And then we want to get a sample of the bacteria onto the inoculating loop. When we do this, we want to do our best not to keep the top of the petri dish or the sample open for too long so that the air doesn't get to that. And before we put the sample on, we do want to put a little drop of water onto the slide. So we will do that in the little black circle. So we'll put a little drop of water, you can put that back. And then here we go. So we're gonna just tip it up diagonally. We're gonna get a little bit of the bacteria, close it back up, and then we're just gonna rub it right into the water itself. Just like that, you can see that we have our sample. Now we're going to go back to the Bunsen burner and hold it over the Bunsen burner for 10 seconds to sterilize it again. And we're gonna pretend that all of that ketchup or our specimen is off, and then this is clean again. And then we have to heat fix this specimen. So what we are going to do for that is we're again have the Bunsen burner, and we are gonna run this slide over the blue flame of the Bunsen burner 3 times. So we're gonna go 1, 2, 3. That's called heat fixing, and that just helps to keep the specimen stuck onto the slide. And that is something that after the specimen and the water droplet that were on there air dry, that is when we go ahead and heat it with the Bunsen burner, wiping it through 3 times. Once we do that, we have our specimen, our smear completed, and we are ready to stain it.